Our next little session is talking about the research initiatives that the Alliance to Cure is funding, a little bit about the other things that we think are on tap and going on in the research community. You heard a lot yesterday already, but there's more to come. And it's also an opportunity for you to ask us some questions. Yeah, and we named it Getting to a Cure, because that's where we're headed. All right, so the first thing I want to do is introduce my illustrious panelists here. We have Holly Blei, who is our clinical research specialist. Holly's role, and she'll be talking here at the mic in a little bit, is to manage our patient registry and pretty much all of the programs that we have that involve recruiting and enrolling patients. So BVMC, you heard about that from Helen Kim yesterday. All of the research roadmap surveys that went out, Holly was the force behind the data analysis, creating the surveys and doing the data analysis. So she is our person for human interactions with patients on a research level. And then Jianbo Hu is here as his title is Industry Relations Director, but he does way more than that at this point. He is our connection really to the scientific community. Some of you know Amy Akers. She has taken a much smaller role. Other things in her life have taken over, so she's down to a very part-time position. She's not here even this conference, and Jianbo has taken up a lot of that slack, but he is also the one who is helping us to identify the best opportunities for moving a cure forward and targeting our efforts financially and also in making the connections to make those things happen, things that might not already be happening within the sphere of the people that come to our meetings all the time. So this is what our treatment pipeline looks like. And to me, this is like looking at a Van Gogh, because going from like my own personal stick figures, which is where we were about 5 years ago. This is so far advanced and so much further than I could have ever imagined us being. We have multiple classes of treatment that are in trials, and I'm going to quickly go down them because I don't think anybody in the last couple of days has done that in a concise way. Not that I'm concise, but we have a group of medicines called Rho kinase inhibitors. They all work in basically the same chemical molecular pathways, but they do slightly different things. All of them should be tried. Atorvastatin, the statin drug that you have all known and heard about for a very long time, being tried as a treatment for CCM, that trial is finally done recruiting. As of last fall, we are now having to wait for the people who were last enrolled in the trial to finish their cycle. I believe it's 2 years of taking the drug before we can find out whether or not it's safe and seems to be somewhat effective. If it seems to be somewhat effective, there are two possible routes that it can go. One is that we could just maybe go to FDA and say, hey, this looks like something, depending on the strength of the effectiveness, this looks like something that could work. Can we maybe expand the label of the drug? That's the least likely. More likely, they're going to try to find funding to expand the trial out to see even more so how effective it is. In the meantime, we have two now companies who are developing Rho kinase inhibitors for CCM. One of them has been around for a very long time. Neurelis. This particular compound started with a company called Bioaxone, whom we brought into the CCM field. They were developing this type of a drug but didn't have an indication for it. And we said, we're here, and we connected them with our mouse researchers. They tested in our mice. It looked good. So now they have licensed it on to a company called Neurelis, who is actually able to take it the next step into trials with people. That drug is currently in phase 1 trials, which means it's being tested in healthy adults. It's been given an investigational new drug approval by the FDA, so it can go on to be tested in adults. They are expecting that by the end of this year, they will be trying to start, they hope to have their act together enough to be testing it in patients. So you can look for a lot of push and a lot of announcements on our part come the end again, this is on them, it's not on me. If they can have it together by the end of the year, we will be announcing that they're recruiting for this trial of a ROK inhibitor, a ROK kinase inhibitor. A second company came on the screen just 2 months ago, maybe even less than 2 months ago, Ovid. Ovid and another company called Graviton are working together to do a more targeted ROK2 inhibitor. You guys don't need to know all the details of the chemistry behind this because I certainly don't, but the idea here is that it's targeting just a narrower pathway to potentially do some good for our lesions. And we are supporting them as well. They are also in phase 1 trials already. They did not have an indication, and they are just now settling on CCM as the first thing that they want to try their medicine on. They did a listening tour during this conference. Some of you may have spoken with some representatives from Ovid who were here just to find out a little bit more about the patient experience, and they are very overwhelmed by, in a good way, by the response that they got and the responsiveness of the folks that they spoke with here. So those two are in phase 1. Phase 2 for Neurelis' drug, probably the end of this year. Phase 2 for the Ovid drug, 2024 is the hope. We'll see how that turns out. Rec 994, well, you all heard about that one yesterday, so I'm not even going to have to go into detail, but we enrolled phase 2. This is huge. Industry-sponsored drug, finished enrolling phase 2. About a year from now, we will have a readout on whether or not the trial can be expanded. And anybody who's in phase 2, there is an extension, so even if you finish your year, if you would like to continue on in the trial, you may do that. And you then will, for sure, if you're on placebo, be randomized into a treatment arm so that you are either getting low or higher dose Rec 994. Then we have some medicines that are stronger medicines. mTOR and PIK3CA are alternate pathways that we think are involved, or they're connected, but that we think are involved in aggressive growth of lesions, the aggressiveness of lesions especially. And so low-dose rapamycin and alpelisib and actually a bunch of other medicines are out there as possibilities for targeting that pathway. And the purpose of that is initially at least to stop lesions from becoming so aggressive. And those are the ones that eventually become surgical. We want to stop all lesions from becoming surgical. Some of the issues we have here are trying to balance the dosing to keep the side effects at the right level, or figure out how to deliver these medicines in ways that don't create as high of a side effect problem. And we can answer questions about that as we move into the panel, because I'm going to throw that over to Jianbo. CD52 is a microRNA 27 target blocker. We are actually funding research, and I'll go into that in a minute, to pursue whether or not that might have a possibility for CCM, and I'm not going to go into detail here, but that's work is happening in mice. Propranolol, small Italian trial was completed for that. Propranolol is a beta blocker used in, for heart indications, but it's also been used for hemangiomas on the skin, and it's being used for AVMs. It's being used for a lot of things that are vascular. So the small Italian trial was complete. It was an early phase 2 trial. The idea now is to expand that out, potentially do one arm of propranolol, one arm of atorvastatin, and then one arm in which you do both. You take both medicines at the same time, and I think that has, in some case studies, anecdotal case studies, shown to actually be a pretty powerful combination. So we'll see if there is funding that they can get in order to make that happen. You heard about the focused ultrasound yesterday, and oh my gosh, that is like blow your mind cool. And we'll see where that goes. They will probably get a significant amount of funding from the government to take that forward. And then low-dose aspirin, I know everyone in this room is going, huh? Because unless you have been really following the literature the way we do, we've always been told, oh my goodness, do not take a blood thinner because you're going to make yourself hemorrhage. Well, there's some new thinking, and happily, folks in Europe are willing to be the ones who find out whether that's true or not, that it's possible that because the blood that flows through the lesions is flowing slowly, it's pretty stagnant there, that blood clots form, and that blood clots are the actual precipitant to hemorrhage. And so if you can reduce the chance of having a blood clot by doing an antiplatelet drug like low-dose aspirin, that you may actually be able to reduce the amount of hemorrhage. And I wasn't here for Kelly Fleming's session. Did she address that yesterday at all? She did. David said she didn't. And not in any detail. So, but that is, as they've done natural history studies looking at people who are taking low-dose aspirin for other indications, they're finding that those folks are no more likely to hemorrhage, and in fact, it does seem to be somewhat protective. It could be the anti-inflammatory properties, it could be the blood thinning properties. All of that is yet to be determined. Claire. Rustam Al-Shahi Salman is the one who's trying to get funding. Yeah. All right, so what are basically you, those of you who have donated so generously to the Alliance, what are you funding? The focused ultrasound work that has been happening, some of that funding has come from us. Some of what Delaney does, the mice, the funding is in part coming from us, and Jianbo can correct me if I'm wrong. There's some other work that's going on that was in one of the posters that you might have caught yesterday morning, having to do with painting tumors. That work is being funded by us. We gave an $87,000 grant to the University of Virginia to continue that focused ultrasound work, and it serves as bridge funding before they get big money from NIH. We are funding nanoparticle formulation of brain-specific drug delivery. So as I mentioned, some of these agents they're actually chemotherapy agents. Alpelisib is one of those. They can cause significant side effects, but if you have to use them systemically, meaning like they go everywhere in your body, but if we can figure out a way to just get them to lesions, then that reduces the amount of side effect that anybody, or maybe even eliminates the amount of side effect that folks would experience. One of the ways to do that is by delivering medicines in these things called nanoparticles, and I think I have another slide about that. We are funding that right now, we think through the University of Pennsylvania. We're still talking with them, but if they won't take on the project, we have alternatives. I mentioned that we are doing that CD52 project, and the way Jianbo wrote it up is cell-cell junction restoration to prevent blood vessel leakiness. And that research is being conducted at Uppsala in Sweden. And we are supporting not just the research, but also the postdoc who's doing the research, who is brilliant and whom we would like to have stay in the CCM field. That's the other thing that we want to do with our funding, is look for talent and try and postdocs often will go off into other things. We don't want them to go off into other things. So the support is in part for the project and in part support for this person. The advantage that Uppsala has, and as does UVA, these are like two of the only now 3 places that we know in the world where they can take our mice and put them in an MRI machine, and then put them in an MRI machine again 2 weeks later, and then 2 weeks later, and see how the lesions change over time. Everywhere else that's doing mice research, they usually either don't MRI them, or if they do MRI them, they have to kill them right away. Because they're not allowed to go back into the mouse facility after they've been out. These other facilities have their MRI machine in the same facility as the mouse, so they can do that. So this is, again, something extra that we get to support. And then finally, we've given a small grant to the University of Rochester, Angela Gladding, to begin doing work on a specific type of gene therapy. We are also right now not funding yet, but certainly working by connecting the right people to develop models of the disease. You sure you don't want to do this one? To develop models of the disease. He's like, no, you could do the slides. I'll just answer the questions. that go beyond animal models. So it's very good. Our mouse models are excellent, but mice are still mice. And so what we can get to it now, and it's not just because of us, but it's just because the science has gotten there, are models of lesions that are developed from stem cells that are much closer to what a lesion actually is, and are embedded in such a way that you can see change in them, as opposed to if they just take a lesion that someone has had removed from their brain, which is essentially at that point dead tissue. These are living lesions, and they can try drugs and see if it makes a difference to the lesion, these organoids. So we are focusing on that. We heard a wonderful paper from the University of Cincinnati, Cincinnati Children's Hospital, where they have developed what is essentially the first organoid model of CCM. We will help them if we need to, but there are other labs working in this area too. Nanoparticle delivery, I mentioned. Now you can see pictures. A lot of the potential treatments for CCM that would be like huge, heavy-duty treatments have significant side effect profiles, and these are all different ways here to deliver medicines. And what we're looking at first are liposome, I believe, or liposome and lipid nanoparticles, organic nanoparticles, in order to deliver medicines, because they're already on the market for other indications. Other potential treatments, mention low-intensity focused ultrasound, both for drug delivery or, as you heard from Delaney yesterday, just all by itself, to try to stop the lesions from developing or in the first place, or stop them from growing if they're already there. And then there's this thing here called tumor treating fields. And this is something that you haven't heard about yet this meeting, because it's kind of our idea, and we are just starting to talk to the researchers doing it. Very much like focused ultrasound, in that there is wave movement involved. In this case, instead of ultrasound, it's electrical wave movement. We think it would be most appropriate for people with spinal lesions, because it requires wearing something. You have to wear, like, as you can see, sort of a patch, and have a little device on you that sends through electrical fields that are sensitized to the specific cells that are in the lesion. So we need to find someone who can figure out what CCM cells are sensitive to that could help in destroying them. And the idea is that the tumor treating fields would destroy the lesions, and so you would wear it as long as you needed it until the lesion was gone. Did I get that right? Pretty much? Okay. All right, and then how do we make funding decisions? Now, we have a different way of making funding decisions than a lot of organizations. The traditional model is people put out a request for proposals. We have $100,000 to give. Send us your ideas for what you want to work on, and then the organization will pick from that and then distribute the funds. We don't do that. What we do is say, this is what we want to get done. So we want to figure out a way to do targeted delivery. We want to figure out a way to make the focused ultrasound thing work, whatever. And we know all the researchers that are out there. We know what their capacity is. We know what they have in their labs. We know what their track record is. We know how much staff they have. This is information that we don't have to go out and ask for, because we know these people. They come to our meetings. We talk to them. Jianbo talks to them really regularly. So we can then, in our evaluation, pick the top 3 and ask just them to tell us what they could do. What can they do now, in the short term, to address this problem, and then negotiate with them how much that could cost, what that experiment could look like. So we're almost contracting more than granting money. This is what we're giving you now, and this is what the timetable is that we expect, and this is what we expect to hear from you. Again, it's a different model that we think is more effective and directed. And then all along the way, we have a role sometimes in crafting the experiments, because we know what we want. And we have the expertise in-house to be able to help shape an experiment so that it reaches a conclusion that has some relevance to the human condition, which is frequently a problem with science. We like relevance to the human condition, not just the mouse condition. And then we will negotiate postdoc funding. We will have points all along the way where, like, if this isn't working at this point, then we're out. Or if it is working, we'll give you even more money. We can do those negotiations. So please know that when you donate to us, you are not just letting us spread money unwisely, that we really, really are thoughtful and have an end goal in mind, which is the cure. All right, it's Holly's turn. You want to come up here, or do you want to do it from there? Okay. So Holly is going to start sharing with you how you can become involved. Although I want to step ahead of her just once and say that patient expert course that I mentioned, definitely, if you haven't already taken it, please go ahead and sign yourself up for it. It's 5 of the best hours you will ever spend.
Okay, so you have probably heard a lot of people mention the patient registry. Some of you may have already joined. However, we just upgraded last November to a brand new registry. The website, I'm going to say if I can, well, it's on our website too, is ccmregistry.org. So even if you are a part of the old one, as we are kind of calling it, we do ask that you sign up on the new one. And signing up on that is an invaluable tool for us. It's a way that we can understand your journey as a CCM patient. It's also a way that you can request free genetic testing. If a lot of questions have been asked during the conference, how can I get testing? How do I know if I'm eligible? Signing up through our registry is the very first step to get that ball rolling. And it also, as these clinical trials come up that Connie mentioned, we can scan through our patient registry to see who might be a good fit for that based off of the study's eligibility criteria. And if you meet those standards, we will personally reach out to you and say, hey, there's a new study that came up that you might be a good fit for. Here's the information. Here's how you can get connected. What questions do you have? So that's a really great way to do that. And thank you. And then by joining our patient registry on the new site, you can also sign up for our biobank. So when you join the registry, how the flow works is you will ask if you agree to allow your information to be housed in our registry. Additionally, do you want to sign up for our biobank? And there is a consent form that kind of goes through the process of what exactly it entails. But basically, it's just a way if you want to provide biological samples at some point. It may be blood, it might be saliva or stool, pretty much anything. But you can let us know if you want to do that by joining up in our registry. You do not have to be a part of that if you don't want to, so you can still join the registry and say no thank you to the biobank if you would like to do that.
All right, so I already kind of went through the trials, but just know that there will be 2 trials coming up that will require enrollment or for which our patients will be eligible to enroll. I do not know the inclusion or exclusion criteria for those trials yet. The companies don't know the inclusion or exclusion criteria for their trials yet. They're still working on that. I do not know where they will be located. The companies do not know where they will be located. But ideally, what happened with Recursion is that we worked very closely with them to come up with inclusion and exclusion criteria, to come up with the clinical endpoints. What are they measuring to see if our patients are better? We also help them to develop the different trial sites. So all of the places, the 15 places that they have open, are mostly the direct result of us having the connection there to help them get in. And they were reimbursing for travel. They continue to reimburse for travel. So if you're in the trial, you don't pay a penny. And in fact, you get a stipend for every visit. It's not much. I'm sorry, there's a bug here. It's like $150 a visit, but it's something. Hopefully it will pay for childcare or time off work. The idea is that your time is valuable and that they want to make sure that you're compensated for it. So when there is an industry trial, there will not be cost to you. And it's important that we participate, that we show up. If we want a cure, we should be part of this. They do test these medicines in healthy volunteers first to make sure that they are not harmful to healthy people. I got to tell you, the first trials are safety trials for our patients, but they would discontinue a trial very quickly if there was an indication that there was any reason to think something was unsafe. And they do check with you every step along the way in ways that are give them more information than what you would be able to just report. So they do blood work, they do urine analysis. I know people who are in the Rec 994 trial sometimes will leave after having given 4 tubes of blood for a variety of tests. They follow up with MRIs. They make sure that you stay safe, because the last thing they want is for somebody to get hurt on their watch. So look for announcements coming up. All right. We are also this year engaging in an update of our clinical care consensus guidelines. Though that is the standard of care. That is where our experts come together and say, this is what the literature says, this is what we think, this is how our patients should be cared for, and it includes sections on genetics, neurology, neurosurgery, radiology, and then just general information about natural history and epidemiology of the disease. I'm the one who's running this project, so I know it's being run well. And we have our literature search already done. We're about to hand it off to the experts in the writing groups, and they can start hashing through and reaching consensus. That's why these are clinical care consensus guidelines, consensus on what good care looks like. And that's all I have for slides. So at this moment, we now have time for questions.
Can I add a
Yes.
quick point on? For the registry, it's especially important for me to go in to look for information that might not be in your mind that's important to the disease, right? So for example, the comorbidity and what kind of medication you are taking. If you are spending time in your registry, please try to put in as detailed information as you are comfortable with, especially what kind of a medication you are taking, at what frequency, for what kind of indications, because we are at the stage looking for all the medications that might be implicated in vascular disease, in heart. So it might be possible that some of the medications might be later on be found to be related to the CCM. So we want to know that.
I'm going to piggyback really quickly. For those of you that were at our family conference in November, where we had just kind of like the bare bones of our new registry, we have now, all of the surveys are available now. So the comorbidity survey that Jianbo mentioned, the medication, those are now on your patient profile. So back in November, you just did the bare minimum with your name and some personal information. If you haven't done so, please log back in and fill out the lengthier surveys, because they are now in waiting for you. That's all.
I just want to make a comment about the clinical trials. And for those of you who haven't heard my comments before, I'm very passionate about this. Clinical trials need to be diverse, which means we need everybody of all ethnicities, all genders, to register for these clinical trials. Because otherwise we don't know if these medications work on men, women, Black people, Asian people, Hispanic people, everybody. So everybody from all backgrounds need to register for these clinical trials. If you have friends who are affected by this illness, who are not here, who are of varying backgrounds, please encourage them to register in our patient database, so that as new drugs enter trials, that if they are a fit for these trials, that we can ask them to enter these trials. And also, if you are wary, like Connie said, they've been cleared and tested before they enter trials. And if anything surfaces that says that, you know, we should stop this, they will stop it. Don't be fearful, because this is what we need to find a cure. This is what we need. So I plead with you, please, please, please. This is my passion, please.
Well said. Thank you. That was very well said. Very well said.
Just have a quick question. Holly, you mentioned the biobank and signing up for that. If we do, is our personal information and identity blinded in that?
Yes. In short, yes.
Is the old biobank the same as the one now?
I can take that.
The old biobank is being transferred to the new biobank. We have, most of the samples in the old biobank are old. And so the tissue has all been distributed at this point. There's no tissue left in the biobank. That tissue is tissue that's been surgically removed through, resected, and people have donated. There's still DNA left in that biobank that we are transferring. So those of you who may still have some residual sample left, those samples will be transferred over, and we will associate anything new that you don't donate with the old samples that are still there. They have been really, really useful. I'm sorry, this little gnat is like driving me crazy here. They've been really useful, and the researchers are now asking for a lot more. They'd like more tissue. They would like plasma samples and serum samples, which require special processing. We did that last family conference. We couldn't pull it off this family conference, but we're going to try to figure out ways to make that happen. I didn't tell you, but they're looking for iPSC lines. They're just like, they want us to be able to provide the things that only patients can provide.
The first set of drugs that you showed, the Ro-K, what are those used for? Are they for stopping lesions, getting rid of lesions, stopping the bleeding?
Right. So the atorvastatin is the first of them that has been tested. And the idea behind atorvastatin, what they're trying it for is to make sure that second hemorrhage doesn't happen. So people who have had a hemorrhage recently get on atorvastatin, and then they're measuring to see whether those people who are on drug are less likely to have a repeat hemorrhage. I do not know exactly what the others are planning. I do know the Ovid folks feel like if you stay on a ROCK inhibitor for a longer period of time, that it will reduce inflammation around the lesion in addition, and so may have longer-term benefits, not just for hemorrhage, but also for the size of the lesions. That doesn't mean that's what's going to happen. They need to test that.
I have a question about the genetic testing. Once I complete my registry, what happens next? How do I sign up for the testing?
So this is another part of what I do, and I love doing this part. So when you go into the registry, you register. If you have multiple lesions that cannot be explained by the presence of a DVA, and you learned what that was just last session, or a history of brain radiation, then you say, I want genetic testing. And it's also if you haven't had a family member tested already, because if you have, you've got the same mutation, sorry, but you do. You will get from me a packet of forms that most of them are for you to fill out, just saying that you agree to be tested and that you agree for the lab to send the result to me. But one of them needs to be signed by your physician. And the physician will need to attest to the fact that you have multiple lesions. And that gets sent back to me. I take all those forms, write up a test requisition, send it over to the lab. The lab prepares a saliva collection kit that gets mailed to your house. So this is like Ancestry, DNA, easy. It gets mailed to your house, you spit, you mail it back, and about 4 weeks later, you will get an email from me saying that your genetic testing report is in and I have sent it to your doctor's office. Please contact your doctor's office for the result. This is the only legal way to do this, and the only ethical way, honestly. I do not want to release a result to you directly without knowing that there's a doctor in the loop. Once you have your result, the index patient, the person who has the lesion, has their result, then any first-degree relative who has not yet been diagnosed but is at risk can get testing through us as well. Same process, though. We need to have a complete set of forms for everyone, a doctor's signature for each individual, but we can do that. We can make that happen.
This is kind of a basic question, but with the trials, how much access would like your neurosurgeon, your primary care physician have? Because I know you have to protect the intellectual property, but then I'd also want my primary care physician, Dr. Steinberg, other people to kind of know enough about the drugs, if I had an incident or just my day-to-day. How much information do people, medical people outside, your medical team outside of the study have access to, if that makes sense?
Yeah, it varies. So here's the thing with a trial, you may be on placebo. So I think the assumption is always that you are on placebo, because if there is an adverse event, they want to treat, they don't want to make the assumption that the drug is going to take care of it. So no matter what happens, you would continue with your clinical care as before. Depending on the site where the trial is, if the site is in the same location that you're getting clinical care, there can be some crosstalk. They can release MRI results, they can release labs back and forth. I don't know how well that works between institutions. So if you're getting your clinical care in, New York and you're in a trial site in Oregon, I'm not sure that that's possible. So clinical care remains separate in most ways. Yes.
So we have minor children. Are they included in the genetic testing?
Mm-hmm.
Okay.
Yeah, we can test minors. And it varies how we do the testing. So if you have children that are under 4, they won't do spit tests. Depending on the genetic result that is found in the parent, we may be able to do swabs. In some cases, though, we need to do blood draws. You would get the appropriate kit sent to you depending on what's needed.
Okay, thank you.
Okay.
My question is on future research. So one area that's of an interest of mine is in nutraceuticals, diet, lifestyle, and based on a lot of the conversations that I've had with other members, that seems to be a common area of interest. I have seen a lot of research really in other areas, cancers, autoimmune diseases, with pretty neurodegenerative diseases with some very interesting data coming out. And I'm wondering, is that an area of interest, or what would it take for that to become an area of interest?
Aside from money? So
Figured that much.
Yeah, funding is a big issue for that. I think, were you there yesterday morning when that question was asked by any chance, when Dr. Kim and Dr. Fleming and Dr. Marchek addressed the question? I'll repeat what they said.
Okay.
So we'll start with, I'll start with Dr. Kim's question, or answer instead, and sort of like reverse it. So the BVMC study is doing a microbiome study as inside of it, but they are also having people do food diaries. They're having just a lot of research is already happening in the context of that study. Now that is enrolling only people with familial disease, but frequently there's no, and in this case too, there is probably no reason to think that the results would be any different for sporadic cases than for familial cases. So there is some of that work happening. It's just still in process. The microbiome, Dr. Kim explained to you that the way the BVMC was funded, it was funded in 5-year cycles, and it's only in the most recent 5-year cycle that microbiome was included, because we didn't know until 2017 that the microbiome may have any implication in this disease. So it's already happening there. The other place that it is happening a little bit is at Mayo with Dr. Fleming. As she mentioned, it's very difficult to do that kind of research because compliance is an issue. If you are telling people, okay, you cannot eat strawberries, what's the first thing you want? You want a strawberry, right? So it's hard to get folks to participate in these trials and know that they are actually doing what they're supposed to, what they've been asked to do, unless you're looking at it in a very controlled short-term way. Other than that, I don't know of research that's happening. So the mouse work that Doug Marchek mentioned was very promising, but the researchers are kind of like feeling that they're at the end of where the mice can take us. So we're in that unfortunate space where there's not enough money for people research, and yet we're kind of done with the mouse research. And that happens, unfortunately, with a lot of drugs, too. It's happening. Vitamin D is another area. And this is, okay, I want to follow this up. Dr. Morcos was asked the question, what can we do to prevent another hemorrhage that, that in order to reduce our chances? And there is an answer to that. There is, make sure your vitamin D levels are at the normal level, at least. Don't walk around with really low vitamin D, you Irish people over there.
We're on it. We're on it.
Okay. Don't walk around with very low vitamin D levels. It's an easy test. It's a blood test you can get at your PCP's office. They might charge you for it, but it's worth it. I mean, seriously, the difference between a $200 blood test versus however many $1,000 brain surgery or hemorrhage. Do that. The anti-inflammatory lifestyle that he talked about is critical. It really is. And so no smoking, get your vaccinations, including your flu vaccination on an annual basis. Just take care of yourself in the ways that will keep you healthier in general. And that is the best thing that you can do to prevent hemorrhage and vitamin D.
Not that these are life-threatening, but I have the skin hemangiomas and lesions on my liver as well. Of the drugs that are in trial, do you know if any of them are, I know you mentioned that there are some that are addressing like the skin hemangiomas, but the ones that are in trial for the brain, do you know if those additionally address the hemangiomas and the lesions in the other parts of the body, which I've been told that the liver, they do think is directly related to the CCM.
Yeah, so I don't know about the liver. I can't comment on that. I do know topical formulations of propranolol have had efficacy in hemangiomas on the skin, so the kind that you see in babies. One of the conversations that has happened in this meeting is, should we be testing topical versions of those drugs on skin lesions first and then seeing if they have any effect there? Can we draw some conclusions about whether they should move forward based on their effect on skin lesions? And so there's going to be a point, probably not that far in the future, where you're going to get a little survey from Holly saying, do you have a skin lesion? Do you think you have a skin lesion? Can you send us a picture of it? And if so, would you be willing to put this cream on it to see if it makes a difference in an obviously controlled way?
This is sad to say, but I had a hemangioma cut off. That was more painful than my brain surgery.
We're not going to take it off. We're gonna ask you to put a cream on it. Oh my goodness. I think Claire is next.
Yeah, sorry. First of all, I want to say thank you so much to all of you for inviting me and my family from Ireland over to this amazing conference. It's been a fabulous 3 days with yourselves, and we've learned a huge amount. I have two questions. The first question actually is for you, Jianbo. Just in relation to the patient registry. So Ross's information is in the patient registry. He has CCM3, as I know we've talked to many of you. I wondered, I haven't actually looked at it recently. I know I just got an email in the past few days, but I do update it as it comes through. Is there a question about the exact gene deletion? Because we have that information, and I don't think that I have included it in the patient registry. And is that helpful? The reason I ask the question is because Alex from the UK was also here. His son is only 2.5, and he made contact with us because Ross's genetic testing was done in a lab in Rotterdam, and his son's genetic testing was only done recently in a lab in Rotterdam, and he contacted the lab to see if there was another person with the same gene deletion. He didn't get our information directly, but he did get the information of Ross's doctor. He contacted me, and of course we were happy to connect. But they were the only two people that have been diagnosed in that lab with exactly the same deletion, 10 years apart, if you know what I'm saying. So I'm just wondering, is that valuable information potentially?
Of course it is. It might not be valuable right now, but if there's like a specific drug targeting that deletion, we can share with our scientists and say, hey, here might be 2 patients that might suit for that drug. And also for our patient recruitment, that also is very important.
And is that a question currently then in the patient registry, exactly?
It's not because people don't know it, but there will be an opportunity to upload medical records. So if you have it in print,
Yes, I do.
then that.
I'll upload it. Okay, perfect. And sorry, one second question then. Just on the biobank, is it possible to contribute to that from an international perspective, or is it mainly US-based?
I don't remember.
So from a consent standpoint, if you join the registry, you will be presented with the consent form to agree or not. I think whether the opportunity arises, I'm not sure, but there is the opportunity to consent or not.
Okay, thank you.
Yeah, for sure, if you come back and we are taking samples at a conference, we can accept samples from you. GDPR is complicated. It complicates everything. Yeah, but if you're here, we won't tell them.
That's perfect. Sounds good.
Hi, I have a question about anti-inflammatory diets. It can be really overwhelming to read up on it. Can you offer maybe 2 places to start, aside from not smoking or alcohol, things like that are kind of obvious? But is there any place to start as to how to get started on anti-inflammatory diet?
Darla, please walk up to the front.
Hi. How to get started. So we have a lot of information on our website about gut health and eating for reducing CCM risks. So have you read that section of our website? I would delve into that first because it's a pretty robust amount of information. And there's some links to some good studies there too. And then we have support groups twice a week. And we talk about this kind of thing a lot. So, if you have specific questions, hopping on there and batting that kind of thing around is helpful. Oh, and Fig. Who uses Fig? It's an app, and I will talk about it more this afternoon, but it's Fig, which stands for Food is Good. Fig.
So she's going to talk about it more, but I'll give you just the highlight is that Fig allows you to scan food and determine whether or not it has ingredients that we have identified as being perhaps not so good, emulsifiers and preservatives, and they have worked with us directly to create that list and incorporate it in the app. And Darla will share more later. For the moment. Are you done with me? Never. Candace.
I want to piggyback on the question about skin lesions.
Yeah.
I know we had discussed this before. One, have they come up with a good example of skin lesions in people of color? And 2, has there been any kind of information presented to primary care physicians or even neurologists that can be used to diagnose or at least ask them to question whether or not seeing skin lesions on individuals can be an early sign to help them at least inquire about whether or not this person could have CCM?
So Dr. Zlotov at the University of New Mexico, and you know the answer to this, Dr. Zlotov at the University of New Mexico has documented skin lesions in Hispanic patients there, and those were the lesions that you saw on the screen from his paper when Dr. Kim presented yesterday. When we started our Breaking Barriers project, which is specific engagement and outreach to Black patients, our support group for that group met and started talking about skin lesions and said, is this, they shared information about the lesions that they saw in their own skin and came back to me and said, what do these look like on Black skin? And we went to the literature, and there was nothing. There is not a single picture of cavernous malformation type lesions on black skin. So we went back to Dr. Zlotov, and his fellow was interested in picking up with this, but we ran out of bandwidth and did not take it any further, and the fellow left and went somewhere else. So as we're now talking about the idea of testing medicines on skin, this is something that is going to have to come up again, and Barrett Zlotov is the person that they're going to be talking with about testing medicines on skin. Part of the problem is photography and getting consistent photography and people being spread out all over the country. So we'll have to figure out how to manage that. All right. Darla is, okay, one last question.
Mine's super quick.
Okay.
You had said Dr. Fleming at Mayo was doing the microbiome. Is it Dr. Fleming or was it Dr. Kim from yesterday? Okay.
Dr. Fleming is a clinician, and she has probably the most extensive clinical database in the country, and I'm pretty sure that she has the capacity to ask the question. She doesn't have food diaries, et cetera. Dr. Kim is in charge of the Brain Vascular Malformations Consortium that has indeed been doing now 4 years worth of work in collecting data. They just haven't gotten enough yet to analyze because it's very hard to get people to give stool samples. Go figure. So that's been the complication there, but they're working on it.